Hello again. This is a second video on Fisher's test of significance, focusing on the implications of using the test. In an earlier video, I talked about the basic workings of the test, which I said it could be reduced to 5 simple steps. Now we are going to look at the implications, and by implications I mean the theoretical or philosophical implications of using the test. Now, Fisher, as I said earlier, is a very flexible tool, easy tool to use. And mostly, although you could have a vague idea of what you want to do and which test you want to run before you do the research, you could pretty much figure this out later on, once you have collected the results and you're ready to analyze them. The implications of this is that Fisher's test is mostly a posteriori research strategy or inferential statistics strategy, which also gives you quite a lot of flexibility. You could, for example, devise new tests after the data, even tests that you didn't think beforehand about using. It's very suitable for ad hoc research, research that doesn't depend on, for example, repeated sampling from the same populations, etc. Therefore, it's also quite suitable for exploratory analysis. And within the framework of frequentist statistics, the statistics based on sampling data and gathering information from evidence from empirical research is a good starting point. And by this I mean when you know very little about the research question you are exploring, etc., Fisher's test is a very good starting point for gathering that evidence. Now, the second step of the test was to set, or mostly assume, the null hypothesis. And the implications of this are that, statistically speaking, the null hypothesis is always true. I mean, for example, we were working with this t-distribution for 30 degrees of freedom, and we said all t-values can be not only be located on that distribution, but also had an associated probability which is dependent on those degrees of freedom that we had that we are using, depending on our sample size. This distribution is true. It's generated so as to say that for that particular t-distribution with those particular degrees of freedom, each t-value has that specific probability. So you cannot deny that. So always the null hypothesis, statistically speaking, is true. On the other hand, we are talking only about the distribution of the null hypothesis. There is no alternative hypothesis, statistically speaking. But this, I mean, there is nothing that alternative hypothesis provides to the test. Nothing we need to take into account. So the alternative hypothesis reduces to being a negation of the null hypothesis. So it's not really a hypothesis at all. I mean, it doesn't count because it doesn't contribute anything to the test. So there's only just one hypothesis that is used for the test, and this is the null hypothesis. Our third step was that once you have the distribution, then you locate your research results on that distribution, just to see how far it is into the details of the distribution, how extreme the result is. So basically, we locate our research results against the theoretical result, the theoretical t-value on that distribution, and we assess, we have an indication, or actually we know the theoretical probability of those values in that distribution. Now, this p-value is also a theoretical probability. I mean, the real probability of the actual research data is 1. It has occurred, you have observed it. So you cannot have a probability of happening because it already happened. So the p-value that you get is actually a theoretical probability of, any t-values in that distribution will have that probability of happening. Now, your actual p-value then is better interpreted as a percentile. For example, we were using, in our example, if you remember well, our p-value was 1.6%. So this p-value can be interpreted as having fallen within the top 1.6% of the potential values in the null distribution. The fourth step was to, once we got that assessment of significance, that location in the distribution, to assess the statistical significance. And we can do that with conventional levels of significance as well. Now, one important consideration to have in mind here is that the null hypothesis really is quite a silly hypothesis. Remember, the distribution is created based on statistics from our samples. So it's quite a null distribution tailored to the particularities of the sample we are working with. Alright? So why is this null hypothesis created? So because it serves as a standard against which to compare our research results, and because this standard is that of 0, of no change or no correlations, etc. It allows us to create that exact distribution for to create that standard comparison distribution. So basically, the null hypothesis is there, or its sole purpose is there to serve as a standard to be rejected. So it doesn't have much value otherwise. Alright. Now, for rejecting this standard, if there is any difference between, for example, between groups, it's very easy to reject the null hypothesis and find a significant difference if your sample size is large enough. Now, how large this sample size can be, we cannot guess, but any difference, even any minimal difference between the two groups, can be found significant if the sample size is large enough. So many times when we find non-significant results, it's because the sample size was not large enough. But that also means, well, then the null hypothesis doesn't have much of a value other than being a standard to be rejected. That's why the p-value serves as evidence against the null hypothesis, because its whole role is to serve as evidence to reject that silly hypothesis we are working with. So basically, not being able to reject the null hypothesis doesn't mean much for the status of the null hypothesis itself. It doesn't tell you much about, for example, that there is no difference. Actually, it tells you more that if there is a difference, you have been not able to capture it because your sample size perhaps was too small. And this may explain why Fisher and some other authors recommend ignoring non-significant results, because not that they may be used as evidence that there is no difference, but because the null hypothesis was silly to start with. So accepting the null hypothesis, if you wish to call it this way, really doesn't tell you much about anything really. And finally, we have the interpretation of results, and that interpretation being as a duality. Either something very rare happens but is still explained by the null hypothesis, or the null hypothesis doesn't explain those research results. Now again, we need to consider that that the null hypothesis has been created as a standard for the particularities of the sample you are using. Therefore, if you get a significant results and you want to generalize, this generalization is to a population of samples similar to your research sample. Basically, the population to which you can generalize your results is inferred after you have gotten the results, or in this case, the significance of your test. So it's not a population that exists beforehand. It's a population you generalize based on the results of the test, on a significant result. And that generalization is therefore limited to the population of samples similar to your research sample. But also, when you look at this, a single research project really doesn't tell you much. I mean, it's hardly evidence of anything, really. How sure can you be that your results are explained by a hypothesis other than the null hypothesis? So, the safest alternative, or the safest way forward, is to replicate the research again. Now, replication is to repeat the research, not necessarily in the same conditions, but kind of in a way that uses a different group, a different sample. Maybe there are some changes in the experimental conditions, etc. But basically, it's still quite similar to what you did before, so that it counts as a replication. And from this replication, you will get again a significant result or a non-significant result. Now, a simple count of whether replication has been significant or not is really not a very suitable strategy under Fisher's approach. Basically because, statistically speaking, it's very difficult to get a significant result, because it depends a lot on your sample size, and of course depends also on the size of the effect, but you don't know the size of the effect. So the best strategy actually is to use meta-analysis, which actually Fisher recommended as well. The meta-analysis that you aggregate when you do the replication, the second study, then you aggregate what you know from the first study with what you got from the second study, which has the benefit of zeroing on on better estimating the real effect size in the population, and also increases the sample size and therefore the sensitiveness of the test. So meta-analysis is a better approach than simply replicating and finding whether the research result is significant or not, the replication is significant or not. And with this we finish also this video about the implications of Fisher's test of significance for better science.